What is the last thing that you people have written in your notebooks? Check and tell me. Also, everyone, every student, switch on your videos quickly. Where are the rest of the students? Switch on your videos quickly. We don't have time to waste. Khadija, Amna, switch on your videos and tell me what is the last thing you people have written. Satika, you tell me, you were there in the last class.
Let me check.
Okay, so today's topic is the ethical issues. Now, see, students, we know that while we are doing the genetic modification of any organism, let it be a lower organism or a higher organism, it is not a very decent behavior if you think about it. Imagine taking a mouse that is living its life normally, happily, and then you are incorporating weird stuff into its body, because of which it can die, it can develop several diseases, and it like its normal life would be hampered. So normally, whenever we work at any place or whenever we like with the community in which we live, right, there are a set of rules that we follow, isn't it? For example, all of us live in a community. You people have your localities, the colony that you live in. For example, you're walking on the road and you'll just go to the person and just give him a tight slap. Is that you can say allowed? Can you do that? No, like you can do that, but that's not the right thing to do, isn't it? Why would you hit somebody else? Or, like just someone is walking on the road and then you push them to the car side, like the part that has traffic in, but that you can't do. So there is like a certain guideline that you people follow, isn't it? There is a certain guideline all of us follow. For example, even if we have cold, do we go to the other person's face and then cough on his or her face or sneeze right in front of somebody? No, we hide ourselves or we cover our mouth or all of those. So this is what ethics mean. Ethics is a set of standards by which we live in a community, right? A harmony community, and that is like when you think about ethics, then you decide what is a legal activity to do and what is illegal activity. Same thing when we bring the ethics into the world of biology, right? While we are doing genetic modifications. Now, in this whole entire chapter, the two chapters, biotechnology, all of you saw how we were, you can say, altering the genome, right? The DNA and the RNA, meaning we are going at the organism level and creating new stuff or like silencing stuff. So basically, that also requires a set of rules and regulations. So that comes under the bioethics. Okay. Now there is the Genetic Engineering Approval Committee, GEAC. This full form is many a times asked in the MCQs. So Genetic Engineering Approval Committee, this is an organization that the Indian government has set up, and this tells us how the activity is valid during the genetic modification research and how it's not. Do you think, like for example, all of the different kinds of uh, biotechnological application and processes that people have studied, did you notice this one thing, that we have studied it only in the prokaryotes mostly, right? We mostly incorporate stuff into the genome of the prokaryotes. Do we do it mostly in the eukaryotes? For example, you are like, you have developed a vaccination or you have developed a particular kind of medicine. Do you just straightly go and inject it into the person to test it out? No, we first do it on the mouse or anything of that sort. So, like, even for the mouse, also, you are disrupting its life. You are basically destroying its life. So, it is a very regulated procedure, and this tells us what is valid and what is not valid. Meaning, you can't do any testing on humans. It's not ethical. You can't do so. And even for the animals that are transgenic, there are set of rules that are to be followed. Okay. So this is what bioethics is related. Then comes the last topic of the chapter, that is patent. Now see, patent, do people understand what is the meaning of patent? Have you ever heard about copyright? What is a copyright? Like, what do you understand by copyright? Tell me in like layman language. I'm not talking it with respect to biology. What is a copyright? I say, do you know copyright? You like. I say, what is a copyright? You like. What? Tell me what is a copyright. See, copyright, for example, or like you might have eaten from KFC, McDonald's, and all of these chains, food chains, right? Now, for example, if you were to open a food chain just like that, but you did not open a franchise, meaning you did not do it from the chain of restaurant or chain of eating joints that are there. You opened your own restaurant, and that is you named it as KFC or McDonald's. Now you're basically stealing the name of the other company, right? That is copyright. Like they can claim you, they can sue you, and then you have to pay the fine. So same thing is patent. For example, if there is like KFC, okay, KFC, that is Kentucky Fried Chicken, right? Then another person opens PFC, that is problematic fried chicken or anything of that sort. Okay, so you use different names, and if you have developed, for example, KFC, when the KFC was developed or when that company was released, the name got patented. Meaning, having a patent means nobody else can use it. So there is no company that can use the name KFC anymore. That's just them. Okay. There can be many, like, in India, like you people might have heard about Burger King. In India, there is Burger Singh. So they did you can say, the technicalities, or you can say the name different, and then they got it. So they'll not be copyrighted, right? But they can't use Burger King because it has already been patented. So anything that has been patented means it has been reserved for that person or for that company. Now, if you try to use it, you'll have to pay them so that you can use it. That is what happens in franchising. I'm taking food chain as an example. Food chain, I'm saying. I'm taking the food companies as an example. But when we apply the same thing to the biotechnology, that comes biopatents. Okay, let us see. A set of exclusive rights granted by a state or a national government to an inventor. Okay, for example, you invented a new word and you got it patented. So now that is your word and nobody else. Nobody else can do that. Or you researched something. You found out why anything, right? And you got that research patented. Now nobody, if anyone else does that same research, it will not be valid because that is yours original content. That is what it is. Okay. So to an inventor or their assignee for a limited period of time in exchange for a public disclosure of an invention. Patents are supposed to satisfy three criteria: novelty, non-obviousness, and utility. Now, novelty implies that innovation must be new. It cannot be a part of prior art or existing knowledge. Non-obviousness implies that it may not be documented, but in otherwise well known. But like it is well known, but it can't be documented. Then the discord fact or product should be of a particular use for the human beings. All of these things, you can say constitute the patent criteria, right? This is what the criteria, like your company, your thing has to be standing on these criteria. Now, a patent is granted for an invention, including a product, an improvement in an earlier invention, or the process of generating a product, concept, or design, right? For example, there has like already been human insulin chemically synthesized. Now you are improving it to make it better, that they can be orally administered. Maybe it's not possible, but maybe. So that is like a improved version of what has already been existing. So you can get a patent for that. Okay. So but at present, patents are being granted for biological entities and for products derived from them. These patents are biopatents. the patents that will be applied to the field of biology. Primarily, industrialized countries like USA, Japan, and members of European Union, they are awarding biopatents to the companies. Now, there is a public growing anger that a certain companies are being granted patents for products and technologies that make the use of genetic material, plants, and other biological resources. Now, when we talk about the patent, there is a story. Like, you know that Asia is famous for utilizing rice as their, you can say, main staple food. Right? Asia, all over Asia, rice is. Now, rice is an important food grain, the presence of which goes back thousands of years in Asia's agricultural history. There are an estimated these many varieties of rice in India alone. Imagine, in India only 2 lakh varieties of rice are there. And the diversity of rice in India is one of the richest in the world. You know that the basmati rice, the aroma, the rice because of it, like which is used to make biryani, right? This is the most like the best rice ever. So this rice has a unique aroma, it has a unique flavor, and it has been developed in India. Apart from that, you know, in the basmati only there are 27 varieties of basmati rices grown in India in different parts, right? So now what happened that earlier, because no one knew what patent was, so what the Britishers or the other countries did once they colonized India, so they saw that okay, India had a variety of rice and it was like very nice and it was like um, what do you say, demanded by the public and everything. So they saw how it was developed. They used that technique to develop the same in their own field. So they basically cheated, copied, and then they got it patent. They got it patent. So India did not have any patent for it, but the other countries that cheated and like what do you call it? Stole the technique. They got it patent. Now India had to pay tax to them, even though the whole entire concept was India. Now you people understand the importance of patency. This is what it is. Okay. Then comes biopiracy. Now, biopiracy is the term used to refer to the use of bioresources by MNCs, that is multinational companies, without the proper authorization from the countries and the people concerned with compensatory payment. Like, for example, India had gotten the patent for rice, let's just say, and if the USA or the other country would have come and used the same variety, now without paying India or without, you can say, appreciating or giving credit, if they would have used that, so that would be biopiracy. Piracy, you understand? You're not from that time, but I don't know if you people have done that. Like many people used to copy or burn CDs. Or like even now in India, like when any new movie is released, there are certain people whenever they go to watch the movie, you know that once the movies are released, they're not available on OTTs, right? Like the television platforms, they're not available there. You have to go to the cinema to watch the movie. So there are some people that go there and record the movie and then they upload it on the internet. Okay, and they earn like that. So that is piracy, right? That is cheating. That shouldn't be done. But the same exact thing when it is done in the field of biotechnology, that is biopiracy. Clear? There's nothing. It's all
What is the difference?
difference between what?
Patent.
Patent and biopiracy.
Yeah.
Patent is when you get your work recognized. Okay. And after you have gotten your work recognized, if somebody else comes and says that I have done it on my own, but you have already patented your thought. You can also patent your thought. For example, if you have thought anything of any research or anything of significance, it's not that I'll eat this today. No, you can't get that patented. But if you patent anything, and if any other person does the same thing, now that person, now you are liable to charge that person to use your thought or use your idea. That is patenting. And if any other person uses your idea, your thought, your technique, and calls it their own, without giving you credit, without giving you money for it. That is biopiracy. Got it? So most of the industrialized nations are rich financially but poor in diversity. Meaning, like they have enough money in the country, but they do not have diversity. They do not have the land, they do not have the soil or anything, and they don't even have the traditional knowledge. In the contrast, the developing country like India and the underdeveloped world, they are rich in biodiversity, they are rich in knowledge, but they do not have the money, they do not have the resources. So, traditional resources include all those organisms that can be used to derive commercial benefits. Traditional knowledge related to bioresources is the knowledge developed by various communities over long periods of history regarding utilization of bioresources, meaning the use of herbs, use of drugs, and everything. In India, it's like time immemorial that people, whenever anyone used to get a cut, they used to apply turmeric on it, haldi, right? And like in our food also, turmeric is used. And it has been used because turmeric, not for color, it has been used because turmeric has anti-cancerous property. That is why we use it in our food. In all Indian households, turmeric is used, haldi is used, right? So now, if you think about the other countries, they don't use turmeric, isn't it? They don't use that. So they don't know the value of turmeric as an anti-cancer product. But now you've been using turmeric in your food because it is anti-cancerous, but now they claim that they use it because it is anti-cancerous. Your value, your thought, your practice has been nothing now because you did not tell the world, you did not got it patented, and you got biopiracy. Like you got under the biopiracy thing. Clear? Now, the Indian Parliament has recently cleared the Second Amendment of the Indian Patents Bill that takes such issues into consideration, including patent terms, emergency provisions, and research and development initiative. So, Indian Patents Bill is there so that these things can be taken into consideration. Okay, these issues can be addressed. Okay. I think this is the lightest part of the whole entire chapter. It's all fun and theory. Now, what you people do, you note it down. I'll tell you I'll tell you the parts that you people note down. This one, in this whole entire paragraph, the whole entire write up, you don't have to write down every single thing. Let me just mark it for you. This part and this part. That's it. You should know. You should write this also and GEAC. Oh, that's above. Okay, only these parts you write and write a little quick.
Done.
Everyone's done?
Yes, ma'am.
Done, students?
Yes, ma'am.
Be quick, students. Be quick. Done, students?
Ma'am, 1 minute. Done, ma'am.
Okay, everyone's done?
Yes.
All right, students. So this chapter is basically over. Now, what I was thinking that before starting on to the new chapter, because only 2 chapters are left, so let us do the revision of the molecular basis of inheritance. Okay. Now, for those of you who want the revision to be done, you can stay in the class. And for those of you who don't, can leave. Mariam, but you will be staying. Okay. What is the difficulty that you're facing?
The whole chapter.
Like particularly?
I just know some topics here and there,
Hmm.
but like, I don't know. I need like a revision of the whole chapter.
See. All right. So when we talk about the molecular basis, there are some terms that you should understand. Do you know what a nucleoside is? What is it?
Nitrogen base.
Not that, not whatever it is.
Nucleo.
Well, give me one example of nucleotide. Okay. Okay. What is the difference between DNA and RNA?
DNA is deoxyribonucleic acid and
But
RNA.
what is the difference?
DNA is double-stranded. RNA is single-stranded.
Yeah, correct. DNA can be double-stranded and single-stranded. Then, see, the difference between DNA and RNA is that DNA is made up of deoxyribose sugar, whereas RNA is made up of ribose sugar. Now, when we talk about a nucleoside, whenever a nitrogenous base, that can be a purine, that can be a pyrimidine. Purine, what are the examples of purine?
Adenine
Adenine
and guanine.
very good. These are the purines, and the cytosine, thymine, and uracil, these are the pyrimidines, right? So when any of like the purines or the pyrimidines, when they form a bond with a pentose sugar, meaning a 5-carbon sugar, the structure that they make is a nucleoside. And when this nucleoside has a phosphate group attached to it, then the structure that it would make will be the nucleotide. Now, in the nucleoside, the nitrogenous base is linked to the OH group of the first carbon of the pentose sugar with a bond called as glycosidic linkage. Right? Because we know that OH is a hydroxyl group and it is present in the sugar area. Right? So at the OH group, the nitrogenous base gets linked and that is through a bond called as N-glycosidic linkage. Now, whenever the phosphate group is attached to it, so it gets attached at the fifth carbon of the nucleoside through the phosphoester bond, right? And whenever two nucleotides are to be linked together, so the phosphoester bond will become phosphodiester bond, and that will form the dinucleotide. These are the basics of the chapter. Now, when many nucleotides are present, it forms a polynucleotide chain. Poly, you already know, more than 2 or more than 5. Now, in a polynucleotide chain, phosphate moiety is present at the 5' end of the sugar. Phosphate group is present at the 5' end and the OH group is present at the 3'. Okay? And you do know that 5' to 3' or 3' to 5' is the polarity of the DNA. This much do you know, right? Now, the backbone of the polynucleotide chain is formed by the sugar and the phosphate. The backbone, like the sugar and the phosphate together, it forms a, you can say, a strong structure for the polynucleotide chain. Because if you remove the sugar or if you remove the phosphate group, the polynucleotide chain will become very weak and it will get destroyed. Okay. And like, for example, like how I've drawn, this is the backbone and projected from it at 90-degree angle will be the nitrogenous base. That can be purine or it can be pyrimidine. That is why when two DNA molecules are linked, we know that A is linked with the T and G is linked with the C. Now, in the case of RNA, now all of these things that we have discussed, it is mostly DNA based. Okay. And our whole entire topic revolves around DNA itself. Now, when we talk about the RNA, that is the ribonucleic acid. So, in them, what happens is that every nucleotide, they have additional OH group. Okay, they have extra OH group present because that same OH group is not present in the DNA. And in the RNA, thymine is not present, instead uracil is present. This is the difference. Okay. Now, talking about the DNA, this is our like main important component of this chapter, the DNA. We know it is a polymer of nucleotides, right? Deoxyribonucleotides. Because here at the second position of the sugar, there will not be any hydroxyl group present at the second position of the sugar. Now, the DNA, it was first identified by Frederick Meister in the year 1869. He was the one who identified the DNA and he named it as nuclei. At that time, he did not know that it was DNA. Then, in the year 1953, Watson and Crick, they gave a proper model for DNA structure. They told how the DNA structure is a double helical structure. And this research of Watson and Crick, it was based on the X-ray diffraction data. X-ray diffraction is a procedure which was produced, like this procedure was done by Morris Wilkins and Rosalind Franklin, like the two scientists, they did this experiment. And based on the result of this experiment, Watson and Crick gave the double helical structure of DNA. Any doubts up until now? Sure. So after that comes the Erwin-Chargaff rule. So Erwin-Chargaff rule, it tells us how the DNA is stable and doesn't matter which species you're talking about, DNA from one species to another will always be stable. Meaning the amount of adenine and the amount of thymine, they'll always pair up and it will be equal amounts. So according to him, purines and pyrimidines, they occur in equal amounts and a purine always pairs up with a pyrimidine. Always. Purine and purine can't pair up. And when you talk about the ratio, so A plus G upon T plus C, meaning adenine plus guanine, that is purine, upon pyrimidine is equal to 1. The ratio is 1, the constant for all the species, meaning they will all have the same amount of purine as well as pyrine. This is what this law says. Clear? Now this ratio, it is less than 1 in the case of eukaryotic organism, that is 0.92. This one ratio, like less than 1 is 0.92 obviously, and it is more than 1 in eukaryotes. Such as in humans, it is 1.52, meaning the ratio of purines to pyrimidine, it becomes 1.52 in us humans and 0.92 in the E. coli. Clear?
Yeah.
Now the features, let us see what are the features of the Watson and Crick model. So basically the DNA, it has two polynucleotide chain that run anti-parallel to each other. Anti-parallel means if one position is 5' to 3', the other will be 3' to 5'. It will always be like that. This is anti-parallel situation. So the DNA, it has two polynucleotide chain, and the backbone you already know is made by the sugar and the phosphate. Now the two chains of DNA, they run in anti-parallel fashion, and this is their polarity. Like the 5' is one pole, the 3' is another pole. So it is the polarity. Clear?
Yes.
We know that a purine forms a bond with pyrimidine, and always remember this bond is hydrogen bond. In between adenine and thymine, hydrogen bond is present, and so does in the case of guanine and cytose. Now the two chains, they are coiled in a right-handed fashion. Okay, meaning it is like this, clockwise. The pitch of the helix. Pitch means this part, the coiling area. This is the pitch. Just like how radius is there for a circle,
Yeah.
same way pitch is there for the DNA. So pitch of the helix will be 3.4 nanometer. 1 nanometer is 10 to the power minus 9 meters, that tiny.
Okay.
And in every turn, like for example, one turn is taken, so 10 base pairs are present in every turn. Okay? 10 base pairs. Yeah. Base pairs means adenine, thymine, guanine, cytosine. Their pairs are present. You were getting confused. What is base pair?
No, I understand.
Okay. Now talking about the distance between the base pairs, so it is 0.34 nanometer. These are the, you can say, the data that you have to learn. Okay. Now, how do you calculate the length of the DNA? For example, if anyone tells you the number of base pair the DNA is having, and you already know the distance between the base pair is 0.34 nanometers. So now if you take the number of base pairs, you multiply with the distance, you'll get the length of it. Okay. So like mostly the known value over here is the distance, right? The distance between the base pair, and that you already know, that is 0.34 nanometers. And if I'll tell you that, okay, there are 20 base pairs. So now you'll multiply 20 into this and you'll get the length of it. Okay.
Yes.
Then comes the central dogma of molecular biology. This was also proposed by Watson and Crick. And they said that informational flow is in one direction. It can't go reverse. That is unidirectional flow of information. What happens here? If you look at this one, like the DNA forms more DNA by the process of replication, right? What is the nature of DNA replication in us? Our DNA replication is in, like, it has which nature?
Um,
Hmm?
Nature means
Yeah, nature means how does a replication take place? Do you remember replication mechanism?
which chapter?
I think it's in this chapter only. Semi-conservative replication happens. Semi-conservative, meaning after the replication has been done, one old strand, one new strand of DNA will be there. Okay. So DNA is synthesized from the DNA. After that, DNA undergoes the process of transcription, which forms the RNA. And then this RNA undergoes the process of translation, which ultimately forms the protein molecule. So this is central dogma. Here you can see there is a unidirectional flow of the information, right? From the DNA to the RNA to the protein, one direction. Then comes the reverse central dogma, which is an exception to the norm. Like central dogma is applicable to all organs, but one obviously exception has to be there. So this is like reverse central dogma. It is an exception. This was reported by Temin and Baltimore, two scientists, and this is called as Teminism because they discovered it. Now, according to them, what happens is that in some viruses, such as retroviruses, they have an enzyme called as reverse transcriptase, because of which they can make DNA from RNA. Meaning, in the case of reverse central dogma, from the RNA, DNA can be made. So now, is it unidirectional?
No.
No, it is bidirectional, and that violates the law. Clear?
Yes.
So this is an exception only found in the viruses, not anywhere else. Got it?
Yes.
Okay. Now, talking about the packaging of DNA. Tell me one thing, why do we need to package the DNA?
Mm,
Why do we need to package it? Tell me.
mm, Because
Because there must be some reason to package it, right?
yeah.
See, what happens is that the DNA that we have, right? It's very, 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 very long. It's 2 meters long. Our cell is very tiny. So in order to fit the DNA, you remember I gave the example of dupatta also in the class. The chunni, that how we fold it and all of that. So basically, fit the DNA inside the nucleus, we have to fold it. We have to package it. Now see, the distance between the two base pair is 0.34 nanometer, and the length of the DNA can be calculated. This is the number of base pair multiplied with the distance between the base pair. That comes out to be 2.2 meters, the length of DNA. Means this is expressed in terms of meters. So the length of the DNA is 2.2 meters, but the nucleus is 10 to the power minus 6 meters. Imagine how tiny. So to fit the DNA, we have to package it nicely. Now, the packaging in the case of prokaryotes, what happens? The DNA we know, like, okay, tell me one thing. What is the difference between prokaryotes and eukaryotes? Please give the right answer.
Prokaryotes, they don't have nuclear membrane, and eukaryotes,
They do.
they do.
Apart from nuclear membrane, is there anything else?
The ATS and 70S.
The cell organelles, the membrane-bound cell organelles are also not found in the prokaryotic organisms. We know that inside our DNA, the nucleus is, sorry, the DNA is present inside our nucleus, right? We have a boundary. But in the case of eukaryotes, since there is no nuclear membrane, so the DNA is scattered throughout the cell.
Yes.
So I wrote Y and you gave the answer. So you are in the safe zone. If you wouldn't have given this answer, Mariam, but you did, so it's okay. Now see, the DNA is a negatively charged molecule. Okay, it's negative charge. So in order to pack it, we have to wrap it around some positive charge protein. Okay, then only it will be like positive and negative, it will form a bond together, right? Because the opposites are attract.
Yep.
So the DNA is held with some proteins that are positively charged in a region called as nucleoid. Now the DNA in nucleoid is organized in large loops held by proteins. Because we know in the case of prokaryotes, since they don't have a nucleus, they have a nucleoid. The area where the DNA is present, that is the nucleoid. But in the case of eukaryotes, the packaging of DNA is very complex, very intricate, you can say. So here the positively charged proteins, they are known as histone proteins. Histone proteins are not present in prokaryotes. They are present in eukaryotes. Okay. Now they are basic proteins, and basic means they are rich in positive charge. Okay. Meaning they have NH2 group present. And they have positive charge, and histones, they are called as basic proteins because they are rich in the basic amino acid, that is lysine and arginine. Okay, now the histones, they are organized to form a unit of 8 molecules called as histone octamer. What happens? Many histone proteins, they come together and form a molecule consisting of 8 histones. We call it as histone octamer. And then to that histone octamer, the DNA starts getting wrapped. Just like how you wrap, you might have you played with a top, Lect,
Yeah. Yeah.
right? So you wrap it around and then you spin it, or Beyblade, just like how it is. So same kind of wrapping of the DNA is done around the protein. You've played with a Lect,
Beyblade.
Beyblade. I was asking about the top. Have you played with the top? I have not played it. Why? How would you have played?
No, but I know what it
Even
is.
I have not played it. So okay. So yeah, so now once the DNA gets wrapped around the protein, that is histone octamer, then that becomes a structure called as nucleosome, meaning DNA wrapped around the histone octamer, we call it as nucleosome, that assembly. Now a typical nucleosome contains 200 base pairs. So imagine in that wrapping, 200 base pairs of the DNA has been wrapped. From the nucleosome, chromatin is formed. Okay, when the DNA is not dividing, then the chromosomes are present in the chromatin form, the tangled form. So from the histone to the histone octamer, then histone octamer gets wrapped around with the DNA and forms the nucleosome. And from the nucleosome, the chromatin is formed. And when the cell decides to divide, from the chromatin, chromosomes are. Chromatin is like this. Chromosomes are like this. Clear? Now, the packaging of chromatin at higher level requires additional set of proteins called as non-histone chromosomal protein. Okay, meaning it is like packaging when it is done even at higher level. This is not something you have to study in depth. You should just know that non-histone chromosomal proteins are required during higher level of pack of chromatin. Now, the chromatin is of two types, euchromatin and heterochromatin. Now you understand the difference between chromatin and chromatid.
Chromosome is the whole thing.
Hmm.
Chromatid is the two of this. Two of this.
Yeah.
That is
Two of this,
chromatid.
two of this what? Do you mean to say that the chromosome is formed by the chromatids?
Yeah.
How? Show me with your hands, fingers.
Chromatid. Chromosome.
Yeah, sort of like this.
Yeah.
And they are joined together in the center with the help of
Kinetochores.
centromere. Centromere has
A
kinetochore.
centromere.
Yeah, good. So that is chromatid. But when the cell is not dividing, you know that the DNA is in a entangled form.
Yeah.
So this is chromatid. Okay. And when the cell decides to divide, the chromatin gets condensed, it forms chromosome. So right now the chromatin can exist in the two stages, euchromatin and heterochromatin. The rheochromatin, these are the ones wherein the chromatin, they are loosely packed. For example, at one place there is this kind of packaging, and at another place there is this kind of packaging. So where there is loosely packaged, it is called as euchromatin. And when you stain it, since they are very sparsely arranged, so when you stain it, it will not stain dark. It will take a little bit of the stain. And remember that the euchromatin region is transcriptionally active, meaning this is the part of the chromatin that will take part in the transcription. Talking about the this thing, heterochromatin, this is closely packed. So it will take up more stain. So it will be darkly stained region, but it will not be active transcriptionally.
Okay.
Meaning it will not take part in transcription. Clear? See, the first case is of heterochromatin, how closely they are packed. And the second case is of euchromatin, how sparse they are packed. All in all, if you see this, the DNA molecule, it starts getting packaged with the help of histone protein. But before that, the histone proteins form histone octamer, right? Meaning 8 of the histone proteins come and form the octamer. And then the DNA wraps around them and slowly forms a nucleosome. Now this nucleosome further forms the chromatin, and then from the chromatin, chromatids are formed and chromosomes. Clear? Whole entire thing is clear up until now?
Yes.
Okay, very good. Talking about the search for genetic material. Now DNA was discovered. And before DNA, RNA was also discovered and proteins were also discovered. But no one knew that what is actually the genetic material. Like for example, reproduction has been going on since time immemorial, even when biology was not developed. So people did not know that it was DNA that was being transferred to the progeny from the parents. So this is what the search for genetic material started. The search started in 1926. The first case is transforming principle. Many a times this can be asked in the exam, right? The transforming principle and the concept about it. Now this was given by Frederick Griffith. Many a times transforming principle can also be referred as Griffith's experiment.
Okay.
So you should know it's talking about transforming principle. Now, in the year 1928, Griffith experimented with the bacteria Streptococcus pneumoniae. What is the shape of this bacteria?
Circle.
They saw miraculous transformation in the bacteria. Transformation, meaning the bacteria got transformed. It got changed somehow. Now, the bacteria, Streptococcus pneumoniae, or you can call it as pneumonococcus, it forms two types of colonies, smooth colonies and rough colonies. Now, the smooth colonies are the ones that cause a disease, and the rough do not. S strain, it is the smooth and shiny because of the mucus coat, and the mucus coat is made up of polysaccharide. So the smooth colonies or the smooth strain of this streptococcus is the one that is shiny, and because of the presence of that coat, it can cause diseases. Okay, so the S strain is the virulent. Virulent means that it will cause the disease. The R strain, it has no mucus coat, so it's not shiny, it's rough, and it does not cause any disease. Now, what he did in the experiment, he used mice. Okay, so biopiracy comes along. So he said that we'll take the S strain, injected into the mice. Now we know that the S strain causes the disease. So now the mice that has the S strain, that will develop pneumonia and the mice will die. When you take another mice and you put the R strain into it, now the mice will live because the R strain is not infective. But then he killed the bacteria. He killed the S strain also. He killed the R strain also. Okay. Now the S strain, if you kill the S strain, that is the virulent strain, if you kill it and now you put it in the mice, now the mice will not die because the bacteria is dead. Isn't it?
Yeah.
Are you getting the thing that I'm saying?
The S strain that he heat killed it before injecting it to the mice.
First, we injected it normally.
Yeah.
We saw that the mice died. Then, to modify the experiment, what we did is that we killed the S strain and then injected
Okay.
to see whether, like, does it do anything after being killed. So the mice lived, the mice did not die. But what he did then, that he took the heat killed S strain and he mixed it with the R strain which was living, and then he injected into the mice, but the mice died, even though the mice shouldn't have died.
Yeah.
Because R strain is the one that does not cause the disease, and S strain is the one that caused the disease, but it is dead. So here, the S strain was discovered, or you can say recovered from the dead mice. So once the mice was, like, the gene study of the mice was done, the strain of the bacteria that was recovered was the S strain instead of the R strain. So then they concluded with that the R strain got transformed. It got changed because of the S strain. So the S strain had something that got into the R strain. They did not still know that it was the DNA, but they just know that it was something. Now, according to him, some transforming principle got transferred from the heat killed S strain to the R strain, because of which now the R strain became S strain. So this must be due to the transfer of genetic material. They still did not know it was DNA. Now, to prove it chemically or to prove it experimentally, biochemical characterization was done to find the nature of the genetic material. That whatever is the genetic material, what is the nature? Is it proteinaceous? Is it amino acid? Anything. So earlier, the genetic material was thought to be proteinaceous. It was thought that, okay, the genetic material will be a protein. Now, in the year 1933 to 44, Oswald Avery, Colin MacLeod, and Macklin McCarthy, they purified biochemicals such as protein, DNA, and RNA from the heat-killed S strain bacteria. Meaning, whatever chemicals that the bacteria had, DNA, RNA, protein, amino acid, they took out all of that. And they said that it was the DNA that was causing the transformation. How? Because if the S strain was treated with protease. We know that protease, they digest the protein. And when they cause the infection, the mice died, even if protein was removed from the S strain. Then RNAs removed the RNA from the S strain, and then also the mice died. Right? Wait. No. What happens is that during the transformation, if the proteases happen to be acting on the strain, then even after the presence of proteases, the mice died, right? Like when the proteases were removed, the mice died. RNA was removed, the mice died. But when the DNA was removed, the mice lived because the S strain was not able to cause the infection. So ultimately, it was said that DNA was the genetic material that was being transferred. Now, the proof that DNA is the genetic material, this was given by Hershey and Chase, Alfred Hershey and Martha Chase in the year 1952. Now, what they did, they worked on the bacteriophages, the viruses that infect the bacteria. So the bacteriophages, they attach to the bacteria and enter the genetic material into the bacteria. So now the bacteria produces viral particles. So Hershey and Chase worked to find out whether it was protein or DNA that entered from the virus into the bacteria. Okay, because they wanted to see that whether it is the genetic material that causes the continuation of life or something else. So they did this experiment wherein they grew the virus on phosphorus and some on the sulfur. Out of phosphorus and sulfur, which one is present in DNA? Phosphorus. Where?
On the fifth.
Yeah. On the fifth what? Fifth floor.
OH of the carbon.
Yeah. So phosphorus is present in the DNA and sulfur will be present in the protein. So they did this experiment. They grew virus on the radioactive phosphorus. So that radioactive phosphorus was incorporated in the DNA. But the protein does not have any phosphorus, so it wasn't incorporated in the protein. So when the virus was grown on the radioactive sulfur, protein was incorporated, but the phosphorus was not. After the experiment, like the experiment has been divided into infection, blending, and centrifugation. So now the bacteriophage, one treated with the radioactive phosphorus, the other treated with the radioactive sulfur. Both of them were allowed to infect the bacteria. Now the bacteria, the blending was done so that the genetic material can be mixed. And now the viral particles were separated by centrifugation. Now they wanted to see in the bacterial cells whether it will be the radioactive phosphorus or radioactive sulfur. That would conclude everything. So they found out that the bacterial cells had radioactive DNA or radioactive phosphorus present in it and not the sulfur. It tells us that it was not protein that was causing the disease. It was not RNA. It was rather what? The DNA. Clear? Okay, so now talking about what are the properties of a genetic material. So the genetic material should be chemically and structurally stable because it has to be transmitted from one generation to another generation. So if it will be unstable, then it will be lost. So it should be stable. It should be able to replicate, meaning make its copies, and it should have a scope of slow mutation. Slowly it should mutate, not fast. And it should be able to express itself in the Mendelian characters, meaning it should follow the law of Mendelian inheritance and all. Clear? Any doubts up until now? All right. So we'll keep it till here only. I think tomorrow I'll get your class schedule for completing the chapter. And your test is on Tuesday, right?
Yeah.
Okay, good. Like you took. It's good that you said that you have a doubt earlier because we have time to clear it. All right.
Can
Now
we
see before
tomorrow? Can we
huh?
complete tomorrow?
I'll try. I'll try to complete it tomorrow. It depends because you know tomorrow is a jam packed. Tomorrow is Sunday, right? No, it's not jam packed. We'll see. I'll take a little time from the normal batch classes also, and I'll take extra class also, and we'll complete. Try to complete, but please study the chapter and so that I'll have to do less minutes.
The whole chapter.
Of course. And I know the whole chapter you won't do, but study at least as much as you can. Meaning, like all of the things that are like replication, transcription, study about those things and come. Like you should know what I'm going to teach. I'll teach you obviously everything, but please be prepared.
Can you send me the notes?
Why? You have the notes?
I didn't write because I was absent. That's why I don't know this chapter.
You were absent.
Yeah, many, like out of 6, 7 classes, I was only present for like 3 or 2.
Okay.
That's why, like, I'm finding it hard.
Then it is justified. Then it is justified because you know, like I do not like it when you know, mostly what happens is that students they don't pay attention in the class, and then during the exam they want
No,
extra classes
like
for that. But
even over here,
okay,
the ones that I attended, I remember like what you said, like
you
the
remember
sigma factor and stuff that I was present in that class when you did
which
that.
one?
Sigma factor and stuff
Rho
like,
factor.
yeah, that.
Okay, all right, no problem with that. Okay, so even if you don't study, it's all right. I'll give you that benefit of the doubt because I was thinking that you were there and still you did not understand.
No, whenever I'm present, I always pay attention to your class. Like, yeah.
Okay, yeah, that is just right. Okay, so I'll try to complete it tomorrow, but I don't think it's possible to complete it tomorrow because this is a very long chapter.
Then we can do it till Monday also. We have time.
Yeah, yeah. So we'll do it till Monday too. Okay, so
Yeah.
so I'll see you tomorrow and we'll discuss this chapter. Try to read a little bit. Okay, I'll
Okay.
give you one task. Learn like read till replication.
Okay.
everything about replication and everything. Okay, and then we'll continue. Okay.
Can you send me the notes then?
Yeah, I'll obviously I'll send you the